Sequencing is the process of determining the precise order of nucleotides, adenine, cytosine, guanine, and thymine, within a DNA molecule. Here's an overview of the science behind DNA sequencing. DNA structure. DNA, deoxyribonucleic acid, is a double-stranded molecule that contains genetic instructions for the development, functioning, growth, and reproduction of all known organisms. Each strand is composed of nucleotide bases, adenine, A, cytosine, C, guanine, G, and thymine, T. Sequencing methods. There are various methods and technologies used for DNA sequencing, each with its principles and applications. Sanger sequencing. Developed in the 1970s, Sanger sequencing involves the synthesis of DNA strands using dideoxynucleotides, ddNTPs, which terminate DNA replication at specific points. The terminated fragments are separated by size to determine the sequence. Next-generation sequencing, NGS technologies, such as Illumina sequencing, use massively parallel sequencing techniques to sequence millions of DNA fragments simultaneously. This enables faster and more cost-effective sequencing of large genomes and multiple samples. Third-generation sequencing. Technologies like PacBio and Oxford Nanopore sequencing directly sequence single DNA molecules in real time, offering long reads that facilitate genome assembly and structural variant detection. Sample preparation. DNA sequencing begins with the extraction of DNA from cells or tissues of interest. The DNA is then fragmented into smaller pieces to facilitate sequencing and library preparation, where adapters are ligated to DNA fragments for amplification and sequencing. Sequencing process. In Sanger sequencing, DNA fragments are separated by capillary electrophoresis based on their sizes, revealing the sequence of nucleotide bases. In NGS and third-generation sequencing, DNA fragments are sequenced in parallel using different chemistries and detection methods. Base calling and data analysis. Following sequencing, raw data is processed to determine the sequence of nucleotides in DNA fragments, a process known as base calling. Bioinformatics tools and algorithms are then used to align, assemble, and analyze the sequenced reads to reconstruct the original DNA sequence. Applications. DNA sequencing has revolutionized biological research, medicine, agriculture, and forensics. Genomics. Studying genomes to understand genetic variation, evolution, and disease mechanisms. Personalized medicine. Identifying genetic mutations linked to diseases for diagnosis, prognosis, and targeted therapies. Agriculture. Improving crop yield and resistance to diseases through genomic selection and breeding. Forensics. DNA profiling for identifying individuals and solving crimes based on genetic evidence. Technological advancements. Advances in DNA sequencing technologies continue to drive innovation, including improvements in sequencing accuracy, read length, throughput, cost-effectiveness, and the ability to sequence complex genomic regions and metagenomes. Future directions. Future research focuses on advancing single-cell sequencing, epigenomics, studying chemical modifications to DNA, understanding the microbiome, and applying DNA sequencing in environmental and ecological studies. DNA sequencing plays a pivotal role in advancing our understanding of genetics, biology, and human health, paving the way for new discoveries and applications in diverse fields.